Hi, Lindsay. How are you?
Hello, I'm fine. How are you?
Excellent. Good to see you.
And you too.
Hello.
Hello.
Some people are saying that there's a passcode required.
There isn't. I've made sure it's off, so they can just try again. Just come straight in.
They've done it. They're through. Cheers. Oh wow, 77. Not bad.
Lindsay, could you send an email around that no password is needed, because I have the same.
And I also, because I do I see my Sanquin colleagues, because maybe they didn't try again.
I'm in, Ellen.
Okay, but the people from the Hla lab, are they there or not?
I have checked the participants, but I didn't see anyone.
No, so I'm afraid they started because they are always perfectly in time. I have to send them an email that it's now working.
So it is the same link, right?
Yeah.
My colleagues are having issues as well. Okay.
Yeah.
I don't know why, because there's no passcode on it.
It asked me for a passcode.
Yeah, I had the same problem. I also was not able to. But...
We're up to 95 now, so we've done something right.
When I clicked on the link in the Outlook, I asked for a passcode. Then I copied it from the flyer, and that one did not give me a passcode. I don't know if that's the difference.
I do not know. I will just.
Celina and Ellen, I propose you start, otherwise we are at risk of running out of time. And then Lindsay can on the background answer the message. And I think Bill has just given a solution. That means use the flyer URL instead of one of the website.
I just have one item. Lindsay, can you confirm that Rob Lewsky was able to join, one of our speakers? He was
I'm here, yes, Celina, I'm here.
Oh, good. Okay. Thank you, Rob. Good. Ellen, would you like to lead? Okay, then I'll take over. Welcome, everyone. Thank you for joining our very first educational session of the Blood Transfusion Genomics Consortium. I would like to remind everyone to please mute yourselves at this point. And I would also like to invite everyone to enter your questions in the chat box during the speakers' talks, and then we'll have some time to address them. I guess I'll move forward and we'll start with our first speaker, who is Willem Ouwehand. He received his PhD from the University of Amsterdam. Currently, he is a professor of experimental hematology at the University of Cambridge, and he holds honorary appointments at Cambridge University Hospitals, NHS Blood and Transplant, University College London Hospitals, and the Wellcome Sanger Institute. He is NIHR Senior Investigator and a Fellow of the Academy of Medical Sciences. His research is on rare diseases with a focus on inherited hemostasis disorders. As a founder of the NIHR Bioresource, Willem has coordinated the NIHR Bioresource whole genome sequencing pilot study for rare diseases for the 100,000 genomes project. Along with Dr. Nicholas Gladel, he founded the Blood Transfusion Genomics Consortium, which is a collaborative on blood cell immunogenetics in 7 countries that he'll be talking to us about today. Thank you, Willem, and welcome.
Sorry I could not switch on the microphone. Welcome everyone with my slides also being on. So let's get my slides going. Question A, can you hear me well from London? And B, can you see my first slide?
We can hear you.
Good, but you're in London, Colin, so that is bad. If you can hear me, let's hope the rest of the world can hear me.
We can't
hear
see
you
your
again.
slides
I
though.
can't see your
No
slides.
slides.
I do
No
not see my
slides?
slide.
No slides.
There they come. Good, so now we should have it all sorted, right? Can you see my first slide?
Yes.
Thanks. So welcome all, and really great to see so many people join this consortium, which we jointly have set up and developed over the past 2 years. So Celina asked me to give you a little background there as much as possible in plain English about the main aim of the consortium. So I think if we look together to the future, it is good to look first back where we are coming from. And I think it is always profound for me to realize, if you go back to the readings of Ronald Fisher, that many of the ground rules in human genetics have originated from blood group genetics. And may I take you to the foreword that Ronald Fisher wrote in the book by Race and Sanger, who were at that time the leading blood group geneticists in England. He said there are 9 usable loci, and he obviously was referring to the positions in our genome where the information was for the ABO blood group, the RH blood groups, and some of the other early identified blood groups in people. I think what is more profound, that in that same foreword, he said in this time that they had no computers, and it was all by a blackboard and a piece of white chalk, that he predicted that in due time, the whole will be surveyed. And I think in coming together today at this first meeting, we are really on this hinge point in history where for the first time we can survey the whole genome and we can try to understand the link between genotype and phenotype, as Fisher so elegantly draw out in this 2-color print at the right, that for those of you who are into the specialty of red cell immunogenetics will fully understand what he wanted to show here with the crossing over as the mechanisms that would lead to the emergence of rare haplotypes that at the moment Thermo Fisher has developed an array for that we hope can detect these rare haplotypes. Before we go into the project itself, I wanted to show the importance, and it's like whether I'm speaking to my fund providers, of doing fundamental science. So much of the improvements that have been made in blood grouping and therefore the increased safety of transfusion have found their source from early work, sometimes 10 to 20 years in advance, by scientists in Cambridge. It was Ronald Fisher who worked with Robert Coombs in 1950 and at the same time with Francis Crick to work out the ground rule of the genetics of the Rh blood groups. And then Coombs developed the so-called indirect antiglobulin test or the cross-match test, which is still used today to provide safe matching of blood between patients and donors. Sometimes they forget that when Cesar Milstein came to Cambridge and developed the monoclonal antibody technology, that it were people like Michael Norberg and Greg Winter, and when I worked in their lab in 1990, there was this buzz going around about the monoclonal antibodies against the blood group A and the monoclonal antibodies against blood group B that was grown up in thousands of liters to be distributed around the world to improve the quality of ABO typing. It was later that people like Nevin Hughes Jones developed the first monoclonal antibodies for RHD typing. standing on the shoulder of giants like we do together, I think pointing to three people all linked to the Sanger Institute. First, Fred Sanger by name, John Sulston, who got the Nobel Prize for deciphering the human genome, or at least a big part of it. And very important, want to highlight the role of Shankar, who is a good colleague of mine, who last week got a prize for his chemistry contribution that underlies the technology that is now globally used by Illumina for next generation sequencing. It is our ability to move from slow to fast reading of the DNA code that allows us together to work on the main aim of the consortium project. And you, so many of you are amazing scientists. So we could ask, why a consortium? And what I show in this time clock of my own last 20 years of my career is how, by working internationally, by sharing data with each other, by having trust between parties, that you do good work to speed up developments about understanding the relationship between genotype and phenotype quicker. And here you see that in the blue period and in the orange period and the green period and the yellow period, I lived in the world of consortia, and I'm so pleased to again, together with Connie Westhoff, giving leadership to the Blood Transfusion Genomics Consortium. And Connie and I want to be very clear that in 24 months, we expect the younger generation to take over. So what is the main aim of the consortium? For those of you who walk the work floor in the blood transfusion services that are part of the consortium, it will not have escaped your attention that the typing of the blood groups, the typing of the platelet groups, and the typing of the tissue types, or also called human erythroid antigens, human platelet antigens, and human leukocyte antigens, often happen in different labs by different people who have their own unique expertise to interpret that particular set of genes in the human genome. And I think the main aim of the consortium is therefore simple. It is to introduce a single unified DNA test, which is affordable for the typing of HEA, HPA, and HLA. And before we leave this slide, I just at the top want to highlight why it is important to be able to type the HEA, HPA, and HLA antigens rapidly, cheaply, and at large scale, because it affects the safety of transfusion of blood and platelets. It is essential about transplantation, about which we will hear more later, and particularly for the red cell blood groups, there is still the disorder of hemolytic disease of the newborn that we need to treat worldwide better and prevent better. So what are blood groups? Blood groups are molecular labels on the cells. There are the ABO and D groups, but there are many more. And it is this test that we developed together that we want to test for all these different blood groups. So that what we can achieve, that we match the blood between the patient and the donor, not only for ABO and D, which causes a lot of problems in many patients, particularly in patients with sickle cell disease, but that we can match for as many blood groups as the blood stocks allow us to do, to reduce the risk that the patient will suffer harm from the transfusion instead of benefit. So I have here and I hope it will play. Now, I think for time I will, now here, no, it's good. But it's a risk. Can you hear this?
We cannot hear.
Here, no,
You can't hear it?
no.
I think then I will skip this movie and we can make it available at the next time. So what is the Blood Transfusion Genomics Consortium? It is a coming together of 7, and some weeks ago, New Zealand has also joined, so actually 8 blood services, together with experts in Boston, particularly Bill Lane at the University at Harvard, and in Cambridge University Hospitals. But as you can see at the left dot on the map, the presence of Thermo Fisher Scientific in this partnership is absolutely essential, as they bring the technology and the knowledge of how to make some of these excellent arrays for the typing of HEA, HPA, and HLA. Who are the people? And many of you are on this call. There's this amazing team of principal investigators that you see at the left, and their alternate principal investigators, or their alter ego, who often in team join our regular BGCA meetings. And then there's a core team of the people who run the Gene Titans, to which I'll come back later, that meets monthly at the BGCR meetings to measure the progress of the activities of the consortium. The first steps of the consortium were made by the founders of the consortium. Very much driven by Bill Lane and by Nick Leddell in Cambridge, they looked at how the typing of thousands of samples from Dutch donors and English donors, how the results that were produced compared to the results that were on the computer systems of the blood services. And without dwelling on this paper that has been published, I think the little pie chart at the bottom says it all. Roughly 100,000 head-to-head comparisons were made between the typing result generated in the blood service to clinical standard and the array inferred type. And there were only 73 discordances on 100,000 comparisons. And a substantial number of the errors were not made by the array, but actually were made in the typing at the blood service, particularly on antigens of the Duffy blood group system. So what is the program of research for the 24 months? And we have kept on track with COVID, and it has been amazing by everyone who is at this meeting, who has contributed to that, that this enormous commitment has been shown. Successfully, we have now up to 8,000 DNA samples in the Central Archive in Cambridge. We are at the cusp of distributing those through the 3 testing laboratories in New York, in Amsterdam, and in London. And there they will genotype those samples to subsequently stream the genotyping data back to Cambridge for analysis by a shared team between Thermo Fisher, Boston with Bill, and with Nick Leddell, to look at the concordance between the genotypes generated on the array versus those that are residing in the computer systems of the blood service. We are positive that by May 2022, we should be getting close to completing our clinical study that will produce the data with which we can engage with the regulators. But let's not forget, we will also submit our work for publication in peer-reviewed journals. I would like to round up that there are 2 arrays. There's the big one, that is 800,000 probes for population genome-wide association studies, and 50,000 of those probes are relevant for transfusion and transplantation. One G-Titan machine can produce 752 samples on this platform. Many of you at this meeting have contributed to make this array slightly better than the one that was published in the Gledel paper, but you see here what this array can do. It can predict gender and ethnicity. It can look at Restless-Legs syndrome and iron reserves in addition to the blood groups, the HLA types and the HPA types. The format that will run in Kansas City, New York for the New York Blood Center is the 384 sample format. There are 50,000 probes that are tailored to transfusion and transplantation. And here you see also that it is not only New York, Amsterdam, and London that have the machine. And here's the Gene Titan that has just been placed in Brisbane in Australia. If you have this setup, you can do roughly 3,000 samples per week. And that would really help blood services to type a large percentage number of their donors. So in concluding, how does the future look like? Blood typer was developed by Connie Westhoff and Bill Lane and is used to take the genotype data from the array to infer the HLA or blood groups and the HPA types. The HLA imputation software is used to do the HLA types, but we see the room for developing of blood matcher, which is a decision support algorithm that helps hospital blood bank staff to make the right choices, and blood stocker, what would help blood services to optimally have a mix of the right type of donations in their central bank for distribution to patients at the right time. So we foresee a future very much predicted by many of you, where the matching of blood will not only be for ABO and D, but also for many of the other highly immunogenic systems. Thank you for listening.
Thank you, Willem. Excellent presentation. Do we have any questions from the audience at this point? Again, I'd like to remind everyone that you can enter your questions in the chat box, and I'd like to remind you as well that you should mute yourselves. We'll have some time at the end then for questions and answers. Oh, we actually do have a question that is related to HLA from Anthony Pauls. What is the accuracy of the HLA imputation?
I think that it is obviously not equivalent to next generation sequencing accuracy. So not being an HLA scientist, I'm confident that we can get a 2-digit resolution, but that will be it. What does that mean, Anthony? I think for clinical purposes, I see therefore room for 2 areas. One, the matching of platelets. Andrea Harmer has suggested to me that the quality of typing is good enough to use the data for that. And 2, it might also be very helpful for disease association studies or disease prediction for HLA loci. I hope that that answers your question, Anthony.
Anthony has a second question. How would this system support patient clinical services?
I think maybe this gets a bit, the first step is to come to MHRA accreditation of the array. And as we have made the first steps in engaging with the MHRA, and we expect or hopeful that by in 2022, we will move in the right direction of getting regulatory approval. I think then every organization in every country should make up its own business plans, whether they want to use this technology or whether they see no role for this technology in their organization.
Willem, are both class 1 and class 2 HLA genes addressed by the array?
I would almost love to just hand over to the HLA experts, but I'm John, I see John Ward, and yes. Andrea is on it. John, do you want to answer?
Yes, the array types for A, B, C, DRB 1, 2, 3, DQ, and DP. So it covers the whole spectrum of the HLA antigens, HLA genes. It's very comprehensive and very accurate as far as we can tell.
Thank you, John.
The next question we have is, how many samples need to be tested to be cost effective?
I think, again, that is a decision to be made country by country. So for those who are really involved, people might have picked up that in the UK, the next step towards precision medicine is the Our Future Health study. And as part of that, we go to type 5 million individuals in the country. So that's 1 in 10 people in the street. And I was today talking with Dr. Sarah Trumpeter at UCLH, It's about how we bring this to the forefront of patient typing, because this array does not only inform blood group matching or HLA typing or HPA matching, but it also provides an opportunity to look at risk scores for the common diseases like breast cancer, prostate cancer, heart diseases, etc. And I think it would maybe be nice to review that area of development, which is very recent indeed.
Thank you, Willem. Any further questions from the audience at this time? Hearing none, we'll move on to our next speaker. It is my pleasure to introduce today Dr. Robert Lewsky, who is currently the head of Division of Hematological Pathology, Medical Director of the HLA Laboratory, and Professor of Pathology at the Queen Elizabeth II Health Sciences Center at Dalhousie University in Halifax, Canada. He's also the Medical and Scientific Director of the HLA Laboratory at Canadian Blood Services in Brampton. Dr. Liwski served as a Program Director of the American Society for Histocompatibility and Immunogenetics, which you probably know as ASHI, Proficiency Testing Committee, as the Chair of the Canadian HLA Laboratory Proficiency Testing Committee, co-chair of the Canadian National HLA Advisory Committee, and is also a consultant to the Brazilian Histocompatibility Association Proficiency Testing Program. Dr. Lewsky also serves as in the ASHI Quarterly Editorial Board since 2017, and he's the Editor-in-Chief since 2021. He serves in the ASHI Board of Directors, and he is the incoming ASHI President-elect. Rob, welcome, and thank you for joining us today.
So much, Celina. Very nice to be here. So I'll give a little bit of an introduction to human leukocyte antigen system. I really have no conflicts of interest for the purpose of this presentation. And I understand that most of the people on this call are blood bankers, transfusion medicine specialists. And so, you know, comparing the HLA and transfusion medicine, you'll quickly realize that even if you didn't spend any time in HLA in the HLA laboratory, you really will understand the concepts that we deal with HLA very quickly, because transfusion, pre-transfusion testing and HLA testing are very similar. So as you know, with transfusion testing, we typically type for AB, NRH antigens, and then transfuse blood that would be ABO, NRH compatible. And similarly, on the HLA, in the HLA world, we perform typing on the patients and donors for the purpose of transplantation, and then we would try to transplant patients with HLA matched donors. Of course, when we talk about deceased donor transplantation for the solid organ transplants, that's often not possible, and patients will get transplants with donors that are mismatched at the HLA genes. And then, of course, in both red cell and HLA worlds, we perform antibody screening. If the antibody screen is positive, we then identify the specificity of the antibody. And then the final test of compatibility would be the cross-match assay. And what I'm trying to highlight is that the principles are very similar, but you know, in the HLA world, the technology has really advanced a lot more in the last couple of decades. All the typing right now is done by molecular technique. In fact, you know, when we look at the proficiency testing, very few labs right now are typing by serology, and it's really more as a backup confirmatory method than anything. For the HLA antibody testing, again, we've moved on from cell-based assay. It's all by solid phase assay using Luminex platform and recombinant HLA antigens that are conjugated to the Luminex beads. And even in cross-match, we no longer use complement-dependent cytotoxicity. Very few labs use that. All of it now is fluorescence-based and flow cytometry cross-match. And so just to give you a flavor of how the antibody identification works, basically you have this Luminex platform with 100 different types of beads. Each one has a different mixture of a couple of fluorescent dyes that allow the instrument to then segregate this or distinguish among these beads. And each bead then is coated with a single recombinant HLA antigens. And we have kits that interrogate class 1 antibodies and class 2 HLA antibodies using this Luminex system, which is composed of 2 lasers. One laser is going to be looking at which bead is passing through, and the other laser will tell you whether there's a fluorescent antibody attached to it. So here's a cartoon with 10 beads with different fluorescent signatures. Each one is coated with a different recombinant HLA antigen, and you have a mixture of these beads, which you then react patient serum with. And if the patient has HLA antibodies that are specific against a particular HLA antigen, they will obviously bind the beads that express that particular antigen. And then we use fluorescent labeled anti-human globulin, anti-IgG, to detect this reaction. And this is the typical histogram that you see. So top panel shows class 1 HLA antigen antibody specificity, and on the bottom panel is the class 2 HLA antigen antibody specificity. Basically, each bar corresponds to a different bead or different HLA antigen. And on the y-axis, you have the fluorescence intensity, mean fluorescence intensity. And so you can not only tell whether there is an antibody targeting certain antigen, but also estimate the strength, the amount, or maybe the affinity of the antibody. And of course, that's complicated. There's lots of factors that determine the fluorescence intensity there. We use this then to determine what antigen patient is sensitized against, and then we can perform, for example, virtual cross-match, where we would try to match donors, obviously, that do not express any of the antigens that the patient is reacting against. We would try to choose donors that have antigens only in the kind of negative range. And what I highlighted here in green circles are patients' own antigens. We always correlate patients' antibody testing with their typing. And so the typing acts as a internal negative control, because we typically don't make antibodies against our own HLA antigens. So again, this can be used to choose donors, for transplantation, which we call virtual cross-match. So, I always wonder, because I am involved in transfusion medicine. I'm a hematopathologist by training, and I do contribute and work in the blood bank. How come we cannot put these different clinical significant antigens on the beads to be able to interrogate these, because it would really simplify antibody identification in the red cell world. And the reason we had to move to this in HLA is because HLA system is extremely complex. You'll see in a second that there's thousands of different alleles that were identified over the years. So it's probably a bit more complex than the red cell antigen system. So let's get into it. Human leukocyte antigen, or HLA, is also known as major histocompatibility complex, or MHC, which refers to these loci in other mammals. In humans, these genes are present on chromosome 6, and they are divided into 3 loci. Class 1 region here contains the classical HLA-A, HLA-C, and HLA-B gene. The class 2 region, you see the DR genes, DQ genes, and DP genes, as well as molecules like DO, DM, TAP, and so on, which function to load peptides into HLA molecules. And then there's a class 3 region right in the middle, which contains some other immunologically active genes, such as tumor necrosis factor gene, heat shock protein, and some of the complement genes. As I alluded to before, HLA, or the major histocompatibility complex, is the most polymorphic region in humans. I keep updating this slide, and I could do it daily, and the numbers would be changing. As of yesterday, we have 22,000 class 1 HLA alleles and over 8,000 class 2 HLA alleles. Now, that's at the DNA level. When you go to the protein level, so how many different proteins can you make out of all these alleles, the numbers goes down to about 13,000 for class 1 and 5,000 for class 2. And this is because some of the alleles are what we call null alleles, which are not expressed. There could be a mutation that puts a stop codon prematurely, or perhaps inactivate a splice site, making the molecule not expressed. But also, some of the differences are in the, let's say, intronic sequences or in the exon, but there are synonymous mutations such that the protein that is expressed is still identical between variety of alleles that encode these proteins. Now, when you go down to, what really the effective polymorphism is, I would say there are probably about 500 different alleles or antigens that are common, that can probably describe about 95% of the population. So the remaining alleles are quite rare, identified in, a family and so on. There's a constant rate of mutation going on in this gene. But still, even if you look at, let's say 450 to 500 different types of alleles, you can imagine that trying to match two unrelated individuals at each locus for both alleles would be statistically pretty difficult to do. And so that's why, as you know, many, when we look for best donors for transplantation, both in solid organ side and stem cell side, the first target is the family members, because that's where you're going to have the highest likelihood of finding a well-matched donor. And this is because the HLA genes are inherited as an entire haplotype, so the whole chromosome. So you'd typically inherit, one set A, C, B, DR, DQ, DP from one of the parents, and the second set from the other parent. So each one of us will have up to 6 different class 1 HLA alleles, and actually up to 12 different class 2, because you see that class 2 molecules are composed of an alpha and a beta chain, and you can form a molecule that is a hybrid between, let's say, a beta chain that was inherited from the mom and an alpha chain inherited from the father, and the other way around. And so here's what the inheritance pattern looks like. Again, I color-coded chromosome for easy understanding of this. So if you have a patient that you're looking to find a well-matched donor, the best bet is to look at the siblings, because there's a quarter chance that any of the siblings are going to be HLA identical. There's 50% chance that they will share half of the HLA genes, and a quarter chance that they will be completely different because they happen to inherit the opposite chromosomes from the parents. And of course, the parents and children are what we call haploidentical. They share half of the genes. Only rarely there would be matches, probably frequency of about 1%, if both parents happen to share a haplotype. HLA also can undergo crossover. So the crossover frequency is about 1% between A and C gene here, or between class 1 and class 2 genes here. And it's actually about 5% for the DP locus, because there is a region that between DQ and DP that is prone to recombination there. So you could get a sibling that's only a 9 out of 10 match, or let's say 8 out of 10 match, because of these recombination events that can happen. So as I mentioned, when you're looking at matching a patient with a donor, within the sibling population, it's a quarter chance of finding a match. With a parent, it's 1 in 100. Once you go to extended family, it's 1 in 10,000 or so. And with unrelated donors, it's more in the order of 1 in a million, and that's if they are in the same ethnic groups. And that's why when we look for unrelated donors for the purpose of stem cell transplantation, we typically have to look to worldwide registries, and there are probably about 35 million type donors around the world with biggest registry in the US, the NMDP registry. And in Canada, we have one match registry, which has about 350,000 registered donors. Now, what are HLA molecules? Well, these are surface glycoproteins. So as I mentioned before, class 1 is composed of a single polymorphic chain, alpha chain, that's non-covalently bound to beta-2 microglobulin. And these molecules function to present pathogenic peptides right there in the peptide binding groove to cytotoxic or CD8 positive T cells. And these molecules are expressed on virtually all nucleated cells in our bodies. Class 2 molecules, on the other hand, are composed of two polymorphic chains, alpha chain and beta chain. And both these chains contribute to the peptide binding groove here, where the pathogenic peptide would bind. These molecules function to present peptides to CD4 positive or helper T cells, and their expression is restricted to professional antigen presenting cells, including dendritic cells, B cells, and macrophages, but they can certainly be induced on other cell types during inflammatory conditions. And so this is a bird's eye view of what class 1 HLA molecule looks like with the peptide sitting in the peptide binding groove. And this is a class 2 HLA molecule with the peptide sitting there between the alpha and the beta chain right there. And this is the structure that T cell receptor recognizes specifically on a T cell, and that then causes activation of T cells that are specifically able to recognize these foreign peptides, and leads to the initiation of adaptive T cell mediated immune responses. Now, when you look at the structure of the HLA molecule, a very interesting pattern emerges. So in pink here are the amino acids that are conserved among different alleles, let's say, of HLA-A molecules. Whereas in yellow are the amino acids that are actually polymorphic. So they differ between different HLA alleles of the HLA locus. And you see that these yellow amino acids surround the peptide. In fact, they make up the peptide binding groove, and they dictate what types of pathogenic peptides based on the amino acid sequence of the peptide will bind a specific HLA molecule. So you see where I'm going with this. This explains why HLA is polygenic, why each one of us has multiple different HLA alleles. That gives us survival advantage as an individual, because it allows us to present a lot broader spectrum of pathogenic peptides to our T cells, and ensures that pathogens are not easily able to evade our immune system. And it also explains the polymorphisms, in different areas of the world, depending on what pathogens were exposed to, those HLA molecules evolved to be able to bind certain peptides from certain pathogens better to protect that population. And that leads to this polymorphism, and also the fact that if you have two individuals from different ethnic groups, the chances of them matching the HLA alleles is extremely low because they've evolved in different areas from that perspective. Now, of course, this polygenicity and polymorphism from the transplant perspective is a nightmare, because these differences, histocompatibility differences between donor and recipient, can cause sensitization and lead to development of both T cell and B cell or antibody responses, and that they in turn can lead to transplant rejection, graft versus host disease, what have you. And just conceptually to show you what this means, if you have a self-HLA molecule that happens to have a particular amino acid polymorphism on an epitope here that makes this allele your allele, if I expose you to a molecule that is different and has different polymorphisms in this area, express different epitopes, our immune system can make antibodies against these epitopes. And of course, as you all know, we are sensitized to these foreign HLA molecules through various sensitizing events, such as transfusion, where we have contaminating donor leukocytes and their HLA antigens sensitizing the host, pregnancy, and previous transplantation. And of course, on the T cell side, T cell receptor in our bodies, certain T cell receptor may be specific against a self HLA molecule with some kind of a foreign viral peptide sitting in it. but that same T cell receptor through molecular mimicry can bind to a foreign HLA molecule with a different allele, which has a certain polymorphism here that, with even a self-peptide or some other peptide may cause the binding and stimulate that particular T cell. And it's a very high frequency reaction. About 5 to 10% of our T cells are able to see these foreign HLA molecule, which is much higher frequency compared to a frequency of T cell receptor that's able to recognize a cognate peptide in the context of self-HLA molecule. And this is why we have to place patients on immunosuppression in the context of transplantation. Otherwise, these reactions are just much too robust and will destroy the donor graft or stem cells. So just to go through the HLA gene structure, again, we mentioned that we have class 1 genes and class 2 genes and molecules. So when you look at the class 1 genes, they have either 7 or 8 exons. The exon 1 codes for the leader peptide, which basically gets cleaved off as soon as the HLA molecule is inserted in the endoplasmic reticulum. The exon 2 and exon 3 code for the alpha 1 and alpha 2 domain, which, as I mentioned before, code for this peptide binding groove. And this is really the business end of the class 1 HLA molecule. That's where T cell receptor binds, that's where peptide binds. This is where majority of clinically significant polymorphisms occur. Now, once you go to exon 4, which codes for alpha 3 domain, which basically binds the CD8 molecule that's associated with the T cell receptor on cytotoxic T cells, now the numbers of polymorphisms start going down. These are more conserved areas of HLA molecules. And then, of course, you have transmembrane domain and the cytoplasmic domain. And again, polymorphisms in these areas typically are not clinically significant because the immune system is not really able to see these, either by antibody binding or by T cell receptor binding to that area. Class 2 HLA molecules, as I mentioned, are composed of 2 chains, the beta chain and alpha chain, and both can be polymorphic, except for DR alpha, which there are only really 2 alleles, 2 different proteins, but DQ alpha and DP alpha are certainly polymorphic. And as I mentioned before, both of these chains make up the peptide binding groove. It's the alpha 1 domain and beta 1 domain that make it up. So as you can see, then it's the exon 2 of the alpha domain here and exon 2 of the beta chain here that will actually make that up. And that's where a lot of the polymorphisms exist. Then exon 3 and other exons, again, they make up more constant portion of HLA molecules. And the differences between alleles and proteins in those areas are going to be typically not clinically significant because they're not really recognized by the immune system in any way. So that then leads to the nomenclature. The nomenclature of HLA genes and alleles has to do really with their structure. So first of all, HLA obviously stands for species. So it's a human leukocyte antigen. The A stands or B or C, that stands for the locus. The star means that the typing for that particular allele was performed by molecular technique. If you don't see a star, we are saying that the typing is done at a serology level, so it's just a phenotype. Then you have basically 4 fields. So each allele can have up to 4 fields in their name, and each field is separated by a colon. And here you will have number of digits. So the first field here, 01. That really corresponds to the old serological nomenclature. So when we're calling antigens A1, A2, A3, A11, and so on. Now, the second field corresponds to really alleles or variants within the same antigen group. And they are all numbered in order of discovery. So O1 would have been discovered before O2, before O3, and so on. And the differences between alleles at that second field denote non-silent mutations, so actual protein differences that actually make a different protein. And so, as you can imagine, those differences must be at an amino acid level, really, and happen in one of the exons, anywhere between exon 1 through 7 or 8 for class 1, or exon 4 through 6 for class 2, depending which chain, which molecule we are talking about. Now, some of these differences will be clinically significant between different alleles, and that's usually when the differences are in exon 2 or exon 3 for class 1. Outside of that, though, typically they are not clinically significant. So the protein is different, but in terms of, let's say, clinical significance and matching between donor recipients, they're not going to be significant, because the differences are in areas outside of this main functional region of the Hla molecule. When we go to the third field, differences between different alleles, this designates what we call silent mutations. So there is a difference at the DNA level, but because of the degenerate coding into amino acids, the amino acid sequence is still the same. And finally, the fourth field is to denote differences between alleles that occur at the intron level. And so again, they don't, these differences don't result in different proteins. And so when you look at the third field and fourth field differences, they are not usually clinically significant. Now, where clinical significance could occur is, let's say, if you have a mutation in the intron that takes out a splice site, and that causes the allele not to be expressed. In that case, though, we would use the modifier N to designate that this is a null allele, to tell you that that difference is clinically significantly different. Other modifiers, L for low expressor. So again, depending on where certain mutation can occur in the Hla molecule, making the expression level low, or S means secreted. Some mutations can occur, for example, taking the splice site out in the intron here before exon 5, such that the only area of the molecule that you're expressing includes the alpha 1, alpha 2, and alpha 3 domain. And so you're not able to place this protein in the membrane, and that Hla molecule is actually secreted from the cell. There are a couple of examples like that. So that's the nomenclature. Now let's talk a little bit about the typing. So right now, in North America, most transplant laboratories will type at all clinically significantly loci that are listed here. As I mentioned, the serological techniques have really been phased out, and now labs are using molecular techniques to perform typing. If only low resolution or kind of antigen level typing is required, the SSP or SSO methods would be sufficient. But if high resolution typing is required, you really need to go into sequencing methods. The sequence-based typing by Sanger sequencing has been the gold standard up until a couple of years ago. But with the introduction of next generation sequencing, many labs are now switching to NGS, and that really gives true allele resolution typing in Hla world. So just to kind of go over some of these techniques and the principle, one of the first molecular methods that were introduced in the Hla world was sequence-specific priming, or SSP, where basically they were designing primers that would bind to the areas of polymorphisms. And these primers typically would be designed to bind to polymorphism within, let's say, exon 2 and exon 3 of class 1 molecules, or exon 2 for class 2 molecules, because that's where most polymorphisms were occurring and most clinically significant polymorphisms were occurring. With this, after the priming reaction would be done, you had to then detect the products of reaction with these different primers by gel electrophoresis. So you can imagine that this was a pretty labor-intensive method. and was also restricting the number of reactions that you could run and then run on the gel. So typically the format would be a 96-well PCR tray. And within the 96 wells, you had to interrogate, this majority of significant polymorphisms within the A, B, C, DR, and DQ locus to be able to distinguish different molecules, different antigens at all of these loci. And this assay would take about 5 to 6 hours to perform, and you could really run it maybe 1 or 2 patient samples at once. And like I said, it really was giving just a low resolution typing that was equivalent to serology. You would need a lot more primers to be able to get higher allele level typing. And in fact, there were trays that would allow you to do that. So you'd first do a low resolution typing and kind of identify what antigen patient had. And then if you wanted to do higher resolution typing, you would have supplementary trays interrogating alleles within certain antigenic group that you already knew patient had. Now, so this method was really going out of favor because of kind of how cumbersome it was. But in the last couple of years, it's really resurged, and that's because of the introduction of real-time PCR technology. So you can use the SSP method or approach, where you have sequence-specific primers based on the polymorphisms. But instead of running a gel, you can now detect the positive reactions using real-time fluorescence approach. And also, instead of running 96-well trays, you can now run a 384 PCR tray. So that really improves the ease of this typing method, the resolution of the typing method. And in fact, this is now probably the most commonly used method for rapid genotyping of, let's say, deceased donors on call, because you can complete the typing within an hour and a half with minimal hands-on time from the technologies. There's no gels, nothing. You basically introduce the DNA into the 384 well tray that already has all the primers in there, and then put it in the real-time PCR machine, and then the computer will give you fluorescence signals, in an hour and a half, and the software is going to be able to analyze and tell you kind of antigen level to maybe intermediate level resolution at all clinically relevant loci. Next technique is called SSO, sequence-specific oligonucleotide probe. So this technique is slightly different. What you would do is design primers that amplify, let's say, the entire exon 2, exon 3, and maybe other exons if you wanted to have higher resolution assay. And these primers would be biotinylated. Once you have that product that contains these exons of interest, you then denature DNA and hybridize it with the same Luminex beads that I mentioned for the antibody detection assay. But in this case, these Luminex beads are actually coated with molecular probes corresponding to different polymorphic areas within the HLA gene. And now, depending on how many molecular probes, how many Luminex beads you have, you will increase the resolution of typing. The initial formats had about 100 beads. Now we can have up to 500 beads. So you can really increase the resolution. So that is and of course, this assay can be multiplexed because, in one well, you can interrogate one patient's DNA across all those different probes. And so you could get relatively high kind of intermediate resolution typing at all clinically relevant loci. And you could do 24 patient samples at the same time and get it done within, let's say, 7 hours. And of course, then, if you want to get higher resolution, you really need to go to sequencing. And again, in the past, we used to use Sanger sequencing methods, but right now, labs are really switching to NGS. And you see, for example, with the particular kit that we are using in the lab, and there's multiple vendors that make NGS kits for HLA. We are actually able to use long-range PCR to prime the entire HLA A, B, C, and DQ alpha 1, DP alpha genes, and most of the gene for the DR locus DP beta locus and DQ beta locus. These are a little bit longer, and so it's difficult to perform long-range PCR against such a long regions. And some of the vendors also created primers that allow for all of these different loci to be primed up in one reaction. So you no longer have to run, separate wells or separate reactions for each locus. And then after this long-range amplification, you perform your fragmentation, adapter ligation, size selection, and after you create your library, you can use either ion torrent system or your Illumina system to perform sequencing. And of course, you could put 384 patients into one tube at the end of the day once you perform your reaction. So these NGS methods are really cost effective. They offer relatively quick turnaround time and ultra high allele level resolution for the Hla loci. So what we were able to type with the SSP, basically just the antigen level, the kind of the first field resolution typing, you could then with SSO techniques, you could get virtually two field resolution with some ambiguities that you may not have been able to resolve. And now really with NGS, you can get full gene, 4 field resolution typing at the allele level. And because of the improvements in technology and, labs, instead of just typing at the exon 2 and 3, really now typing the full gene, we are recognizing more and more alleles. And you see that it's just growing exponentially, from, when I started looking at Hla as a resident, my first year residency as a hematopathologist, where we had maybe 1,000 class 1 and 700 class 2 alleles, we now have about 22,000 class 1 and 8,000 class 2 alleles described. Now, why was this pressure to develop this? Well, because this is actually clinically significant. The typing and then matching patients and donors actually makes a clinically significant difference. Again, when I started training in Hla back in 2003, the testing and matching for the purpose of stem cell transplantation was really pretty simple. We would type patients at AB and and DRB1, and the AB typing was done by serology, DR by SSP at the low resolution, and then we would type up all the siblings, and we're really hoping for a sibling match. And if there was a match at the class 1 level, then we would quickly type the sibling at DR to confirm a full ABDR match and go to transplantation. If we didn't find a sibling or there was no sibling, we would actually go to do a search based on this low resolution typing and find donors that were matching at low resolution pretty much, and then move to transplant. But of course, studies since then have showed that this is really not a great approach, because matching in unrelated donor transplantation has to be done at allele level. Otherwise, the outcomes are really poor. And this was the first study, Lee et al., that really showed that. So they showed that matching of patients and unrelated donors was important, that all these loci, A, B, C, and DR. And you see that mismatching at a single locus meant about 15% difference in survival after transplantation, and mismatching at another locus was really detrimental to survival. But also, this was the first study to show that it was actually important to match the donor and recipient at the allele level and not just at the antigen level. If they were matched at the antigen level but not allele level, the outcomes were just as poor as if they were completely mismatched at the antigen level. What does that mean? Is that in the past, if we had a recipient typed at low resolution and 3 donors typed at low resolution like this, this donor here is mismatched at the antigen level, and we would obviously say that this is not the greatest donor. But both donor 2 and donor 3 would be considered well matched, 8 out of 8 matched donors. But now that we perform typing at the high resolution level, you see that in fact donor 3 is not a great match. because even though they match the antigen here, the O4 at the DR, it's a different allele of that DR4 antigen. This one is O401 and this one is O402. And in fact, that makes this donor just as bad as donor 1 really, and donor 2 is the only donor that is well matched in this particular scenario. And as the typing technology and requirements got better and took this into consideration, you see that now the survival of patients that, let's say, have a leukemia who receive a transplant from matched related donor, sibling donor, versus unrelated donor, they are now really identical. And that's because both are now matched at allele level resolution. So I'll finish just by going through a couple of cases just to show you how we work up our patients in the HLA laboratory. So this is the first case of a patient with aplastic anemia, and this patient had a lot of siblings. So we would just type at low resolution, then we would type the siblings at low resolution as well. And of course, we would try to find a match. And I'm sure that all of you are seeing that sibling 4 is a perfect match. We would also perform a family study just to make sure that we were able to account for all haplotypes. And basically, to do that, you look at the, let's say, patient and a sibling that share half of the HLA genes. And now you see that the 1, 8, 7, 15, 6 antigens are in common, but the other antigens are not. So they must have been inherited, the ones in green from the same parent, and the other ones must have been inherited from the other parent, but opposite chromosomes. And so we can now color code these, and already there we have 3 familiar haplotypes, and then we would look to sibling 2 and 3 to find the other haplotype. Once we have the haplotypes figured out, we can actually express the typing instead of just in numerical order. We can express it as inherited haplotypes, and we can also predict what the parents would have been based on this typing. Now, if we have a patient who has no siblings, we would right off the bat perform high resolution typing. And then what we would do is determine whether the frequency of that particular genotype is common or not, which would then help us determine whether we'll be able to find an unrelated donor or not. And so to do that, we can use variety of software. For example, Haplostat, which is available through NMDP, has frequencies of genotypes in different ethnic groups. So we would simply plug in our patient's typing at high resolution into the software, and then it will tell us that this typing is quite common. 7 in a million individuals would have them. That doesn't sound like common, but knowing that, for example, NMDP has 17 million donors available, you know that for this particular patient, you'll be able to find a match quite easily if you go and search through the registries. And in fact, we're able to find a patient or donor for this patient very easily. Here's another example where we have a patient with acute myeloid leukemia and no siblings. So again, we would perform high resolution typing and go back to Haplostats to determine the frequency of this particular genotype. And in this particular case, actually, we find that this genotype is very rare, 1.5 in a billion. So we would not be able to find a perfect match. But can we find a donor that's mismatched for 1 antigens? And in fact, the answer is yes. And again, we can do analysis to determine how likely that is. And for example, if we just mismatch the A0206 for A2501, so it would be antigen level mismatch, we would then find what we call 9 out of 10 matched donor with an A locus mismatch. And in fact, in this particular case, we found 3 donors for this patient by searching registry. Each one, as predicted, was mismatched at the A locus as shown here. What we need to do now is, because we're going to go for a transplant with a potential mismatched donor, is ensure that the patient doesn't have preformed antibodies against these mismatched antigens. And again, so we would go back to this Luminex, this single antigen assay, to measure antibody and detect antibody in patient serum And you see that in this particular case, the patient has antibodies against A1 antigen, which is a mismatched antigen with one of the donors, but not A29 or A25, which are the mismatched antigens with the other donors. And we would basically say that that donor 3 that has A1 antigen would not be recommended, but either donor 1 or donor 2 would be acceptable donors because they have a single antigen mismatch. So I'll stop here. And if there are any questions, I'll be happy to answer. Thank you so much.
Okay. Thank you very much, Dr. Lewsky, Robert Lewsky, for this highly educational talk, informative and educational talk as well. I think most of us are from the red cell field, which is much more complicated than you are aware of, because we have also many more antigens than only those you thought about. And maybe I have some questions. I first have to say to everyone that unfortunately our third speaker will not be, he just had a sudden, we got an email that he was not able because of acute health problems. So, well, it's very sorry that we don't have this talk today. I think we will ask him to give his presentation on the HLA typing using the array another time. So that's also, it's no problem that we are a little bit over time. Maybe we can first start with the questions I've seen in the chat. What is in the chat? I think there is a question from Colin. Now that you have NGS in place, do you still use low resolution typing for family studies?
So that depends on the laboratory. We do not. We've actually, once we implemented NGS technique, we are now typing everybody, whether they need high resolution or not, by NGS. And the reason is that this makes it a lot more cost effective. Because whether you run, 5 samples or 90 samples, you'll use the same chip for the sequencing reaction. So it's a lot more cost effective if you can run more samples at the same time. Now, we still use low resolution techniques for, let's say, rapid typing. If I have to type a deceased donor, I cannot spend 3 days just to wait for the NGS result. Also, we use low resolution techniques like SSO for verification typing. So on a stem cell transplant recipient and donors, we always have to perform typing just like in blood bank, ABO, you have to confirm it on 2 separate samples. So what we do in our laboratory is we do NGS on one of the samples, and then we use SSO on the second sample, just because we're still, kind of learning about the technique and make sure that there's no dropouts or anything like that. So it's very helpful to confirm it using a second method. But other than that, we are basically using NGS for everyone. And honestly, even if you don't need what we thought in solid organ transplant, we always thought that we don't need high resolution typing, that low resolution is enough. But in fact, that is not true. A lot of patients have allele specific antibodies. Right now, everybody is going into describing epitopes on Hla molecules, and actually concept of epitope matching, which probably many of you are familiar with it, because that's also useful for platelet Hla matching. And so having high resolution information and being able to predict the exact epitopes that the molecules have and what are the differences between the donor and recipients at the epitope level is very helpful. So most labs are going to be switching to typing everybody at high resolution, I think.
Yeah, thank you. I think clear answer. Colin, you are happy with the answer, I assume?
Yes, that's fine, thanks. Yeah, and we do this, take the same approach and just NGS most of ours, because I think it also provides information for the future as well, because we're not sure what some of these other polymorphins might mean in the future. So it's good to have the data.
Okay, and then there is a question from Willem, which is maybe similar also a question from Lily and also to Willem. So the question is, what is the chance for a patient of African ethnicity to have an HLA match?
Yeah, it is much lower depending on the ethnic group. I can actually try to find a quickly slide here to show you what the probabilities are for different ethnic groups. Oh. It's not in this presentation. Oh, there it is. Can you see this slide?
Yes.
So at the top are, white Europeans. And here you have several African ethnic groups. So you have, African, Black, Caribbean, African American. So, the probability is more like 20% to find a matched unrelated donor. And this is why, haploidentical transplantation from either siblings that share half of the genes or parents or children or even cousins has really taken off. And with the high dose cyclophosphamide regimen that was developed by the Johns Hopkins, that's really taken over and very useful for ethnic minorities, and same with the cord blood transplantation, where the degree of mismatch between donor and recipient can be a lot higher and can be tolerated a lot better.
What do you think if now our array is implemented, which means that hopefully at one moment all blood donors all over the world will be HLA typed? What would be the impact on this matched likelihood? What do you think?
That's a good question. Because again, with the advent of haploidentical transplantation, where we have where we can just take a family member who matches half of the gene, the HLA genes, and the outcomes are just as good or maybe even better, because the risk of chronic graft versus host disease is actually lower with this. I am not sure whether this is going to be of great benefit. And the logistics around identifying the donor, collecting the donor are a lot easier when you have a family member that can donate compared to an unrelated donor. And COVID really also put that to the forefront, where for some time, because of COVID and all the restrictions, it was very difficult to actually come up with a unrelated donor that was willing to proceed to donation.
So your expectation is that we will totally switch to haploidentical?
Not sure totally. The other area of research now is whether you can apply the high dose cyclophosphamide treatment to multiple mismatch unrelated donors. So kind of a haploidentical but unrelated. And the answer is probably yes. Now, what that will mean is that you'll be able to accept a lot more unrelated donors. They will not have to be as well matched as currently, where you can accept 8 out of 8 or 7 out of 8. And I didn't go into this, but DP matching is also important. So the DP mismatch between donor and recipient is okay, but they have to be what we call permissive. So the donor and recipient molecule have to be within the similar antigenic group. But all that to say is that you probably will have more acceptable donors if you're able to tolerate a larger mismatch. And so then the benefit of having these huge registries, it may not be as important.
Yes, well,
but again, that's just my opinion and seeing a little bit of a trend, but I could be completely wrong.
Okay, but it's good to hear. And are there more questions? maybe I have also another question for you. Is do you think that if we show with the array that we are totally 100% accurate for all the common alleles, might that replace high resolution typing for those common alleles? Do you think that if it's reaching the same accuracy, that the array could replace, at least for the common alleles, which are 95% of all the donors, or white European donors?
So replace it for the purposes of?
Even for stem cell transplantation.
Yeah, that is, I would have, yeah, we'll see what the data shows. I think as we mentioned, you know, having a full allele level resolution is beneficial. We are learning more and more about other areas of Hla molecules that are important, you know, because there's a lot of siRNAs in there and other factors that could potentially contribute to graft versus host or rejection. So with NGS, you know, and sequencing being so affordable, I think that the higher the resolution, the better. So I'm not sure whether taking kind of a step back and providing lower resolution result would suffice at this stage.
So I'll say, at least from my perspective, I think one of the large benefits of the number of people that will get an array typing. It'll allow us to screen very large populations, to then follow up with the higher resolution testing. When we're talking about costs, you know, I don't, Willem hasn't mentioned the cost, but we're talking vanishingly cheaper than current HLA next generation sequencing typing for the entire array, from nuts to bolts. So it will definitely allow us to get millions of more people into a screening and know their HLA at a fairly moderate resolution, at least, to then follow up, I think.
Mm-hmm. Yeah.
Also, I think it's a useful screening tool for finding platelet donors. We haven't talked about platelet donors, but
no, that's of course very clear.
It's a very cheap and quick way of finding platelet blood
Yeah,
donors.
we could likely proceed just from the array to platelet donation matching.
And for platelet donation, again, where there you really do not need, you know, high resolution typing. I mean, intermediate resolution and kind of understanding the epitopes would be sufficient for sure.
Yes.
Yeah, I think the two-field typing provided by the array will allow you to establish the epitopes without taking it further. Yeah.
So,
yeah, and again, you know, if you consider haploidentical transplantation now, but obviously that's for related individuals, then again the array would definitely be sufficient.
Yes.
Robert, could I ask a question, Willem here? John brought up the issue of platelet panels, and I fully agree that the platelet panels can benefit further by typing more donors. But we haven't spoken about disease association. It's partially because I'm rather poorly informed, except that the GWASs that have been involved in HLA always pops up, obviously. But is disease association as a diagnostic test still used, or do you say no, it has been trends and people don't ask for it anymore?
No, we still do this. You know, the B27 for ankylosing spondylitis, DQ2, DQ8 for celiac disease. We do B57 testing for abacavir and others. So certainly it's still something that we do. It is a relatively low volume test, I would say. We may, you know, get I don't know, maybe 1% of our volume is for disease association.
That's really helpful to hear, because obviously the array genotype is of a good enough accuracy to be used for, if you look to Bycroft paper, I think that nails it down very nicely actually. But you say, Willem, it's a very small volume in most laboratory HLA laboratories. Yeah. Thanks.
If Martin Howell, if I could just add with regard to provision of HLA selected blood products, I mean, there's a growing interest in provision of HLA matched red cells to avoid sensitizing organ transplant patients. And I think this level of resolution of the array may be the only reasonable or an optimal way of being able to provide large, or at least have large numbers of HLA typed red cell donors available without massive investment using traditional techniques.
You know, so that's an interesting point, but you also have to worry about probably transfusion associated graft versus host. Then, if you start matching red cell donors with the patients, would you get into trouble with that potentially?
I think those products are irradiated before we issue them.
Yeah.
Well, but then you're going to have to irradiate a lot of products.
Well, no, it's not prophylactically. It's radiated before use. So, we do provide that on a limited scale at the moment, but just for live transplants at the moment. But we are looking into feasibility of doing that on a larger scale.
I guess what I'm wondering though is in patients that currently would not be at risk for transfusion associated graft versus host, could that potentially change if you were always transfusing with HLA matched red cells?
Yeah, it's not something we would do. It's really looking at the renal lists and for live transplants at the moment. So if we know the HLA type of the proposed donor, we would avoid those mismatches so we don't sensitize them before the transplant.
Okay, so it can be selected situations.
Yes, yeah.
I think we are coming to the end of the meeting. Maybe I have to give the word back to Willem, or shall I end the meeting?
No, not at all. Celina and you have organized this, and we would like to thank you for that.
Yes,
so
well,
over to you to close it. Yeah.
Yeah. Okay. Maybe you can stop sharing your screen. Okay. Yes. And well, I think it is very nice to see that there were so many people who logged in to this educational meeting. So I really think people would. Well, it's encouraging to see the interest in this, and it was a good decision to start this, and we will continue. And well, I think we have to apologize again for Alexander Ditley, who he was just not able to give his presentation, and I think we will keep it for the next meeting, because we do not have set a subject for the next time yet. Maybe there's a question about HPA. I think that would also be. Well, I think we also could do. We have discussion, so we have to. We will not decide now. But if you have an idea, just mail it to the consortium, and we can discuss it in a next meeting. And all ideas are very much welcomed, of course. And I would like to thank everyone, and especially Celina, because she has done most of the work, and our speakers. I think this was a very informative meeting today. And I don't know if we have recorded it, because we are in different time zones, because this is just such an international consortium. So Willem, do you know the people in Australia, New Zealand, China?
I do see that, as always, Australia is online at the moment.
Okay.
But I think that it is good what you said, that we should make it available to New Zealand and Australia, and Lindsay will take care that that is taken forward. Yeah.
Okay. Are there anyone who would like to ask something or to say something? If not, then I would, well, thank you all for your attention, and thanks again the speakers. Okay. Have a good day, evening. Yeah. Bye.
Bye-bye.